So welcome to High Rounds. Thank you for coming today. We're glad to have Dr. Scott Letendre here. He is an infectious disease specialist who cares for people at the Owen Clinic. He earned his medical degree from Georgetown University School of Medicine. He completed residency training at the Naval Medical Center here in San Diego, and subsequently completed a fellowship in infectious diseases at Duke University, as well as a fellow in neurologic HIV research here at the UCSD School of Medicine. He specializes in understanding the effects of chronic viral infections such as HIV and Hep C on the brain. His research examines the effects of HIV and antiretroviral therapy on brain health, including cognition, memory, and cerebral vascular disease. He's also interested in understanding the aging process among patients who have been on antiretroviral therapy. We love Scott because he's always willing to do a talk on neuro AIDS, and he is a lover of dogs, especially Vishlas. So thank you. Go ahead, Scott.
Thank you. I like to hear myself talk. So these are my disclosures. So just really almost gone fund from Merck to do a CSF doravirine project. The funds went to Buffalo to one of my trainees there, and then subcontracted to us. So those of you who were at the AETC talk would have seen this, but this is just a reconfiguring of an expansion of an old diagram they used to show. But it's meant to highlight a couple of things. One is the complexity of all the factors that can or could potentially affect the brain in people with HIV, and even people without HIV who have HIV risk behaviors, transmission or risky risk behavior, and that the contributions to neuropsychiatric disorders changes over time depending on the circumstances and the stage of care. So this starts out, as I said before, infection. We've been learning that even people who have risk behavior and with PrEP, I think Andy Cates has pointed out to me before, maybe Jeff did, that people really aren't at risk for HIV, so it's not HIV risk behavior, but it's still risky sexual practices that put people at risk for sexually transmitted infections, drug use and addiction that can affect their brain, and other factors. So, and including social and structural factors that put people at medical risk. So, and the entire field is paying more attention to these. Unfortunately, when people do get infected, they have these factors. There may be some enrichment for drug use and for sexually transmitted infections. People who get more of those have more opportunities to get infected, unfortunately. And so those may contribute more to what happens in the brain after someone does get infected. And of course, after they do get infected, then we have the problems of HIV replication, immune suppression, and neurotoxic HIV transcripts and proteins. And then we're doing such a better job, thanks to the work of many of you, in getting people linked to care and getting them on antiretroviral therapy. And so once they do, we have to factor in potential antiretroviral toxicity into this equation then, but the influence of HIV replication is eliminated, but there's still problems with production of viral products like transcripts and proteins that can adversely affect the nervous system. And then past immune suppression seems to be important as well. And we can treat hepatitis C and control hepatitis B mostly, so the effects of those are lessened. And so we enter this period of clinical stability. People are generally doing well. Maybe they're back to work. Maybe they get their addiction under control. And then we're dealing with this issue of premature aging. And as all of you know, we start to see these age-related comorbidities in people with HIV at an earlier age and to a greater extent. And traveling along with those are use of more concomitant medications than people without HIV. And so we're struggling with polypharmacy. So I'll touch on several of these points as we go along. I got to give a talk recently with a wonderful man, Andrea Calcagno, an infectious disease doctor from the University of Torino. And he showed this slide, and I asked him for permission to show it. And he reminded me that he took it from me. So I've got my own aging issues to deal with. But this is, I'm going to talk a little bit about viral load, some viral load data that we've been generating, we and others. And just reminding you that biomarkers are challenging. And with something as complex as what I just showed you, we're probably not going to find just one biomarker that's going to diagnose, divide people into having their brain more affected by HIV versus having their brain less affected by HIV. And so, but I won't read through this list, but a lot of different categories of conditions can affect the brain. This is an update on a slide I've shown before, and the part I've shown before is on the left. This is work by Paula Desplats and others using brain tissue from people who had a terminal illness with HIV, volunteered in the California Neuro HIV Tissue Network here at UCSD. And after they died, we collected brain tissue, and scientists like Dr. Desplats can use it to study these conditions. And so she measured DNA, RNA, and P24, so DNA, RNA, and a protein, an HIV protein. And when Paula was kind enough to share this manuscript with us, she was particularly interested in this group of people with very high levels of virus, but I was particularly impressed with this group of controllers here who had almost no detectable DNA, no detectable RNA, and essentially undetectable levels of proteins. So, it tells us that there are some people who don't control HIV very well in the nervous system, and other people who control it very well, and so in the brain. And so I think this warrants additional investigation, and we've been trying to do a little bit in that. You've seen maybe a version of this before. I've been working with one of our trainees who came to us from Dr. Calcagno in Torino. He's been here for almost a year now, Mattia Trunfio. And Mattia's took data from our database, 2,000 CSF plasma pairs from almost 600 adults, all of them when they reported not taking antiretroviral therapy. So many of these viral loads are from the early 2000s to, sort of 2010 range. Many more people, of course, now are taking suppressive antiretroviral therapy. In the blue here is the CSF viral loads, red is plasma. You can see that the CSF viral loads are shifted leftward here, on average about 1.5 log copies per mil lower in CSF than in blood. But interestingly, almost a quarter of people have undetectable CSF viral loads using a 50 cutoff when they're detectable in blood and off of therapy. And about 4%, which is the percent that we see CSF viral escape in, have CSF levels greater than plasma. So Mattia's has finished a manuscript, and we've circulated it to co-authors. But there's several of these correlates that Mattia found in sort of expanding our data set to include things that had not been looked at before. So some of these things have been reported before: CSF white blood cells, plasma viral load. No one will be surprised at that. You might be a little surprised, it hasn't been reported before, that people had a higher CD8 T cell percent when they had a higher CSF viral load. No one really had reported on CSF serum albumin ratio, an indicator of blood-brain barrier integrity either. The blood-brain barrier is more leaky when you have a higher CSF viral load. So also not surprising. We actually reported before. So some of these people everyone was off therapy. Some were naive, some had taken therapy before, and people who had taken 3-TC before actually had a lower CSF viral load. And we'd actually reported that before with a trainee of Davies, George Hightower, who went to Duke to do a surgery residency and is done now with that. It was that long ago. But we think this might reflect a reduced fitness of virus, although, drug resistance mutations should revert to wild type over time, and lower serum triglycerides and lower MCV. So we've been interested in this idea of iron metabolism contributing to central nervous system complications as well, working with someone named Asha Kalyampur in Cleveland, who's a fantastic hem-onc specialist. So several new indicators. Can we use this in clinic? Probably not, but at least with all the people we see with a very high CD8 T cell number or percent, we might, be a little more mindful of whether or not that increases their risk for having more virus in the central nervous system. Now, when people are suppressed, then we can't use the usual assay, and Serena Sputich, working with the ACTG, published this analysis a few years ago, measuring cell-free HIV RNA, cell-associated CA HIV RNA, and cell-associated HIV DNA, and showing that it really seemed to be the cell-associated HIV DNA that was most frequently detectable in people who were on suppressive therapy in CSF. And working with Kevin Robertson, before he passed away, of course, they found that people who had detectable cell-associated HIV DNA had worse neurocognitive performance. So, there's this debate about whether or not neuropsychiatric complications in people with HIV who are on suppressive therapy is really due to HIV or is due to other factors. And some people strongly argue it's not due to HIV at all. I think both can happen, and so the problem is that if you just measure a viral load in CSF and it's suppressed and you conclude that there's no HIV effect, you're not really looking at the right measure. Another one of my trainees, Bert Anderson at Emory, we've published several things on using a single copy assay for HIV's RNA, so cell-free HIV RNA. We've tended to see it present more in CSF than Dr. Sputich's analysis. And in this publication, we did see associations with global neuropsychological performance, as well as in these subdomains: processing speed, recall, so memory and motor function. So we tend to see something there, but this was a longitudinal analysis and it did decline over time. So if you look out here, it's maybe not dissimilar in terms of just the percent of people who had it to what Serena found. But we were able to look at this longitudinally rather than cross-sectionally. I did want to also mention this very nice work that was just published, work from the Last Gift program, which Sarah Janella and Davey Smith lead and is linked to the California Neuro-HIV Tissue Network. And what Davey and Sarah did is, a lot of amazing things. But one of the things that they did was enhance what CNTN, California Neuro-AIDS Tissue Network, did in terms of rapid autopsy. So, we try to perform our autopsies in CNTN within 24 hours, but, Davey and Sarah realized that, and we realized that that's not good enough for some sorts of investigations. So they do a rapid autopsy. They bring people in, they follow them very closely as they approach death, for example, in hospice, and some of the participants schedule their passing, and so that they know exactly when it's going to happen. They take them to collect their tissues very, very quickly, and what that does is it enables neuroscientists to take the tissue and do what these scientists did, which was isolate cells. So they actually use this freshly collected tissue to isolate microglia, and then try to address this question that's also been around forever about whether or not the cells in the brain really do harbor inducible DNA that's capable of being induced to replicate HIV. And so they isolated and grew these microglia. They verified that they were microglia and that they were pure cultures. They measured total DNA, integrated DNA, and HIV RNA. And this is in cortex in black and basal ganglia in blue from different donors. And you can see there's DNA and integrated DNA, as well as a lower amount of HIV RNA. But they then exposed the cells to latency reversing agents, and they were able to induce cell-free HIV RNA with these latency reversing agents, with these cells from the brain, these microglia. And then they also did viral outgrowth, and they were able to grow out virus as well. And they showed that that virus could infect other cells. So this was published, it was a lot of work. It was published in the Journal of Clinical Investigation, which is, a really, really great journal that requires this level of multi-stage work. And it's really beautiful, but it really does answer this question that, that HIV can be induced in microglia in people who are on suppressive therapy, in this case at the time of death. So using the single copy assay, Bert and I just reported at CROI some unexpected findings. So we divided people again, everyone was on suppressive therapy, and this was in the context of the Charter Aging Project, but we divided people into whether or not they had basically undetectable, less than 1 copy per mil in CSF versus more than 1 copy. And there was, a substantial association between CSF and blood. There was, in the paired blood specimens, there were only almost nearly all of them were in agreement, were concordant. When you were undetectable in CSF, you were undetectable in blood. Only about 20% or less actually were discordant. But what we did was we compared it to some of the biomarkers that we measured, and the direction, I kept looking and re-looking at this because the direction is opposite of what we would think. We thought that having low-level HIV RNA, a viral product, would be bad for the central nervous system. But all of these indicators, these biomarkers, point in the other direction. So this biomarker, 8-hydroxydeoxyguanosine. Don't expect anyone to know what that is, but it's a nucleic acid product of is a product of nucleic acid oxidation. And so these people who are, sort of suppressed at or below 1 copy per mil, we can't detect any virus, they had more nucleic acid oxidation than the people who had a little bit of virus around. They also had higher levels of this neuronal axonal protein tau. So this indicates more axonal injury. And so again, it was the people who were fully suppressed, if you will, and they had lower amyloid beta, which is the pathologic direction. So there's something about, maybe a little bit of virus stimulates an immune response and you have a one type of injury. But if you're fully suppressed, maybe the effort it takes for the body to fully suppress the virus from immune pressure and pharmacologic pressure causes problems as well. So it's not good news. And I'll come back to my Eeyore sort of not good news personality a little bit later in the presentation. And we didn't find a difference here in neuropsychological performance, but we've been looking more at depression because we're interested in the overall neuropsychiatric picture, as I'll come back to. And these people who are fully suppressed had more depressive symptoms than the people who weren't. So Bert and I tried to understand this, and we said, well, maybe it's got something to do with the medications they were taking. And in fact, we found that the people who were fully suppressed were more likely to be on TAF. And so here just shows CSF, they're more likely to be suppressed less than 1 copy in CSF and in blood. And also, if you were on TAF, you had higher levels of this other neuronal protein, neurofilament light, which is also not so good, although this difference, while statistically significant, is not nearly as large as these ones. So, maybe a little signal that there's something up with TAF. And So why might that be? Well, I was interested in seeing this work from Nebraska, from Kim Scarzi's group there, and people who are switching from TDF, which is a higher dose of tenofovir, to TAF, which is a lower dose, and it's a lower dose, as we all know, because the alafenamide is cleaved at the level of the target cell rather than at the gut epithelium. So TAF achieves higher concentrations of tenofovir diphosphate intracellularly compared with TDF. I just was surprised to see that even with the reduced dose, that this increased 2.5-fold. So much higher concentrations of tenofovir diphosphate inside the cell, which better inhibits HIV. But that's not, we deal with off-target effects of medications all the time. And so this could have effects on the mitochondria. It could have effects on telomeres. So telomerase is a reverse transcriptase. There's data I'm not showing you here from cell culture showing that tenofovir is one of the better inhibitors of telomerase. So if it inhibits telomerase, it can't extend telomeres. We would expect shorter telomeres, which protect chromosomes from injury, from damage. And in fact, in this analysis from our sustained training and aging and HIV research group, STAR, a training program we have, our STAR scholars showed this at CROI last year. People who were on TAF had greater reduction in their telomere length in the Charter aging study than people who were on TDF at both visits versus people who were on other NRTIs. And then at CROI this year, one of our new STAR scholars from Washington University in St. Louis, using data from the WISE study of over 1,500 women with HIV, showed that the TAF-containing regimens, particularly those in combination with either cobicistat, in combination with cobicistat, had more depressive symptoms. So you can see here TAF, Cobi, darunavir, TAF, Cobi, elvitegravir, TAF, Cobi, darunavir again here. So these women had more depressive symptoms than women who were taking other regimens. So, and interestingly here, if they were taking TDF, they had fewer depressive symptoms. So, this is not an overly compelling case against TAF. It just tells us that there are several signals here that maybe need to be investigated further.
Scott,
so,
can I
yeah.
interrupt you for one? Can you go back 2 slides? One more. So Shannon asked a quick question, actually one forward. What was the CSF viral load range in the greater than 1 group?
All of these people had less than 50 copies. They were all on suppressive therapy.
Okay.
So I don't recall using the EasyQ assay, the single copy assay, what the range was, but we did have one person, even though they were less than 50 on the commercial assay, they came back with more than 50 on the EasyQ assay. So we eliminated them from the analysis.
Okay, great. Thanks.
All right. So in Charter, everyone hopefully remembers CNS HIV antiretroviral therapy effects research project's been going on for 20 plus years now. And it's a 6-site study in the US, 2 on the East Coast, 2 in the middle of the country, and 2 on the West Coast. But, we have, for all that time, divided people into the degree of their other neuropsychiatric comorbidities. And I just wanted to show everyone, because we don't always talk about this, if the 15% of people were categorized as having severe neuropsychiatric comorbidities, as we know from seeing our patients, these things are not inconsequential. Brain trauma, cerebrovascular events, seizure history, systemic medical conditions that can affect the brain, substance use, and so on. And they're most common in this group. They're intermediately common in this group, and they're least common in this group. But even in this group, 41% have cognitive impairment and 27% have a systemic medical condition that could contribute to these conditions. So, and we also measure things like cognitive symptoms and activities of daily living, instrumental activities of daily living. So I'm going to come back to that. But, we have high unemployment in charter and in some groups of our patients as well. So that's an issue as well. Oops. So on the heels of that, we just recently published the primary results of the charter aging study. We brought people back after 12 years at these 6 sites, 402 people assessed 12 years apart. So some studies, someone requisitioned our data and published some of them. And at the maximum, I think they had 12 years, but the average was like 6. Everyone here had 12 years of follow-up. And Dr. Heaton developed this global change score that uses neuropsychological testing and data from people without HIV to help interpret what we're seeing. And the good news is that 6% of people improve, 70% remain stable, but about a quarter of people declined over 12 years. And I've just summarized the conditions that we found either at baseline 12 years earlier or at the time of the 12-year assessment that were associated with cognitive decline. And you can see having more depressive symptoms. We keep seeing a signal for this recently as well, chronic pulmonary disease and diabetes at 12 years. Lifetime cannabis use disorder at both time points came up as well, which we were a little surprised about. And then lower hematocrit and having pre-frail or frail status. And these are just a few graphs that show this. You can see at baseline that global mean T-score, an indicator of overall neuropsychological performance, was similar between the 2 groups. But in the non-decliners, it got a little bit better over 12 years. But the substantial decline in the 23.7% who declined at 12 years. And you can see that again here if we just look at it as impaired or non-impaired, this substantial difference. And what we did was we took these conditions either at baseline here or at 12 years. And I'm sorry, this is the 12-year data, either with global change score or whether they declined or not. But you can see the more of these conditions someone had, the more decline they had, and the more likely they were to be in the declining group. So some things to watch out for. We also looked at treatments for these things. So we looked at whether someone was taking a bronchodilator. We looked at whether people, when they reported diabetes, if they were on a diabetic medication, and similarly for hypertension as well. And we did see some indication that people who had hypertension at baseline, when they came back, if they were not on an antihypertensive medication, they were more likely to decline. But in the multivariable analysis, that didn't bear out. What did bear out was that if people had COPD and they weren't on a bronchodilator, they were more likely to decline. So some indications, of course, that treating these conditions is important. Now, we have a Center for Medicinal Cannabis Research here too, and they were not entirely pleased to see that that using cannabis was bad for you, but those people had a cannabis use disorder as diagnosed by DSM-5. And so those people are usually heavy daily users of cannabis. And too much of a good thing is not necessarily a good thing. We see indications that moderate use does have benefits, but this is something else that was published this year by Jeff Rogers, one of our postdocs. And I just thought I'd share it with you. It's from our TMARC study, which is ended now, Translational Methamphetamine AIDS Research Center. But we are trying yet again to get a new center funded. We'll be putting in an application in January, and we'll be bothering some of you about that, trying to get some referrals. But what Jeff showed, looking at our old data, was combining methamphetamine dependence with cannabis use, was that people who had methamphetamine dependence, but who did not use cannabis, here in the blue, had lower, that is worse, T-scores and a higher probability of impairment. But it was in those, not those exact people, but in methamphetamine users, if they use cannabis, it seemed to have benefits for their cognition. So sorry, I'm a little colorblind. So in the red here, you can see people who are meth users who use cannabis. better T-scores, lower probability of impairment. And so if you have some injury, something that affects the vasculature and the immune response, then maybe using moderate amounts of cannabis has cognitive benefits, but more work needs to be done there. We have another active project funded by NIH looking at cannabis and antiretroviral drug-drug interactions. And so we'll appreciate referrals for that. The enrollment has been a bit slower than we had hoped, but these are findings from our our second progress report that we recently submitted. And so cannabis, like any drug, is metabolized and eliminated. Just to remember to make this point, it can inhibit cytochrome P4, or does inhibit cytochrome P453A, as well as UGT1A1, which we know are involved in metabolism of many of the antiretrovirals that we prescribe. So our hypothesis was that cannabis users would have higher concentrations of UGT and maybe CYP metabolized drugs. So here we're looking at dolutegravir. So thank you to Brooke Henry and Jeremiah Momper and the others who are involved in this project. But so far, this is just a very preliminary findings. We are seeing higher concentrations of dolutegravir in plasma in the frequent users than in the infrequent or non-users, because cannabis has some anti-inflammatory effects, and reducing inflammation reduces blood-brain barrier permeability. We also hypothesized that we would see a lower CSF plasma dolutegravir ratio, and you can see, in fact, that is what we seem to be seeing so far. But again, sparse data at this point. Much more needs to be done, but I thought you'd find them interesting. Now, everyone probably remembers we have a negative pressure room here at the HNRC, ABRC, CMCR. It's a mouthful. And we can administer cannabis. So we're taking a subgroup of people who consent, who are cannabis users already. We're bringing them in. We're giving them either high THC cannabis, high cannabidiol cannabis, or low THC, low cannabidiol cannabis, so placebo essentially, and then giving them an observed dose of the antiretrovirals they're already on. And this is just our first person who enrolled. The arms are blinded to protect the innocent. And so we don't know which arm is which here, but this is the same person at two different treatment periods. In one treatment period, they have low levels of dolutegravir, and I meant to look up what the population values of these should be. So sorry, Shannon, I forgot to do that, but maybe you would know, or Lucas, or anyone else who's on. But in this other arm, markedly higher levels of dolutegravir. So again, we're giving them an observed dose. We're giving them either placebo or a form of cannabis vaporized, and then we're collecting blood 6 times or 8 times over 6 hours. So something seems to be happening here, but again, just one person. Don't make too much of these data. More to come, but I thought you'd find that interesting. To add to the complexity of all this, I just wanted to remind you too that participant characteristics do vary substantially by region. So these are the 6 charter sites, and I've just color coded these from better to worse, better in green, worse in red. If we believe that being older is worse. So feel free to come at me for that, but I'm older. So, but just to show how variable these characteristics are at the 6 charter sites. And so we're interested in looking at them by site, but we generally combine all of these data together. But it's not surprising that there would be some differences in studies that are from Europe versus studies that are in New York versus studies in California. And so the findings can be different, but that could reflect the findings could be different, but that could reflect differences in the study population. So just draw your attention to major depressive disorder, neurocognitive impairment, and lifetime substance use diagnosis, and then also show you the different substances that are used at these, mostly at these sites. So no one will be surprised, except maybe that's not high enough, that we have problems with methamphetamine in San Diego. But our participants also use a lot of cannabis, whereas only 24% of people in Baltimore use cannabis, but they're much more likely to use cocaine there and in New York, and also alcohol, whereas we still have a fair amount of alcohol here, but less opioids in San Diego and in the middle of the country versus the East Coast. So these are all different drugs and have different effects on the brain. And so, again, it's a complexity that we try to account for, but is challenging. And so, talking about multi-site studies, I thought I'd show you this other recent publication from the ACTG that was led by Serena Sputich and Kevin Robertson and myself and many others. But it's a clinical trial in which we intensified people's antiretroviral therapy to either add dual placebo, dolutegravir plus maraviroc placebo, or dolutegravir and maraviroc together. And the idea was if the cognitive impairment that we see in people with HIV was due to ongoing low-level HIV replication, production of viruses, because the concentrations of many of the drugs are lower in the central nervous system than they are elsewhere in the body, that intensifying therapy might help. And we designed this before any of the data on the neurotoxicity of dolutegravir came out, but we didn't really see evidence of neurotoxicity, as I'll mention in a moment. So all of these people, 191, had cognitive impairment and HIV, and were on suppressive antiretroviral therapy. And again, once they consented, we randomized them to one of these three arms, and we followed them for 96 weeks. And this graph shows their overall cognitive performance. And there's something called practice. If people are capable of learning, when we give them the same tests over time, they get better. And so we try to alternate versions of the test to try to account for that. But you can see all three arms here. Each of these different colors is a different one of the three arms, but you can see all three arms got better and then leveled out over 96 weeks, and there's no difference between the arms. So intensifying therapy didn't seem to result in improvement in cognitive performance. And interestingly, we don't always see this, but their depressive symptoms, this is the Beck Depression Inventory, their depressive symptoms improved over time as well, but also did not differ between the arms. So I'm going to come back to this because I've already been talking for 39 minutes here, and I've got more to show you. But what those findings suggest is that, again, it's not HIV replication, but that doesn't mean it couldn't be HIV RNA transcripts, just pieces of RNA, or HIV proteins that they're still foreign material that the immune system reacts to in the nervous system. And so Bruce Brew, who some of you may know his name, he's been in the neuro-HIV field since the beginning, amazing scientist and a wonderful person. He's been working with someone named Kazuo Suzuki, who helped develop a method called the double R assay, which is a highly sensitive assay capable of measuring HIV RNA at lower levels than some other assays, so they argue. But remember Dr. Sputich's findings on cell-associated HIV RNA being so low. Dr. Suzuki found it in 87.5% of CSF specimens, and it was even higher than in PBMCs in blood. And this may be because activated cells tend to have higher levels, and activated cells are more likely to migrate into the central nervous system, into CSF. So these typically include mostly activated cells, and there's a lot of cells in the blood. Some of them are activated, some of them are not. So this may be just selecting for activated cells, but regardless, he found higher levels of cell-associated HIV RNA in CSF and correlated it on magnetic resonance spectroscopy with cerebral metabolites in frontal white matter, parietal cortex, and deep gray matter, the caudate. And you can see those levels, these associations here. So this is just the standardized beta, meaning a correlation coefficient, basically. So the lower the number here, the stronger the effect size, larger the effect size. But I don't want to dwell on that too long, again, in the interest of time. But Avi Nath has been, putting forward hypotheses about HIV TAT being neurotoxic and being produced in CSF, and he has been working on an assay for a long time. The assay is very difficult, but they finally succeeded, and they found it in 37% of people of CSF specimens. And he was nice enough to request some of our specimens. And you can see here, he looked at in our specimens, people before and after they went on suppressive antiretroviral therapy. And one person had this dramatic decline in TAT and CSF, but these other people had a modest increase after going on therapy. So it supports that this could happen. It was associated with a few factors, but particularly with use of stimulants, amphetamines or cocaine. And they did find associations with information processing speed and psychomotor function, motor function. So a couple of the psychological domains that we look at. So again, antiretroviral therapy does not, our current antiretroviral therapy does not alter production of just transcripts and protein. It inhibits production of viruses. And so this, the findings from the ACDG clinical trial 5324 I just showed you, in combination with this, suggests that it's not low-level HIV replication that's causing the problem, but these other factors could still be contributing. Now, for clinical trials of therapy, I put this depressing slide together recently for my talk with Andrea Calcagno, but just to show the extent of our failure to find much of anything that's beneficial for cognitive impairment. There was a little signal for Deprenyl many years ago. Ned Sachter, years ago, before he passed away, did some really beautiful work on paroxetine from cell culture to animals to humans, and showed that paroxetine improved global performance. But it also has anticholinergic effects, and nothing's really moved forward since then. But there's also some switch studies as well. And I just want to point out these non-pharmacologic studies. Perhaps the signal is most consistent here for the non-pharmacologic therapy. So, calling out Dr. Lee and all of his incredible efforts with Keeley meditation, Freudian. So Keeley meditation, but also cognitive training and exercise as well. And I thought I just showed this to call out our friend Christine Erlinson at Colorado, who does so much important work on aging and frailty and exercise. And this is just a nice graphic that I won't walk through in the interest of time from one of her articles. But thought I'd show you this nice analysis from Croy, in which they, a group in Spain, followed people for 12 weeks of an integrated exercise protocol. And they showed that aerobic endurance improved, not surprisingly, in people after 12 weeks of this exercise regimen. But why I'm mentioning it is they also looked at anxiety symptoms and depressive symptoms, and these improved as well, as, particularly in people who were more adherent to the exercise regimen. So they divided people who were more adherent and less adherent. And I would talk with Christine recently at Croy, actually, and asked her, how she funded people's involvement in exercise. And she mentioned that I think the county that Denver is in is Denver County, but someone can correct me. But the county, or maybe the state, actually, I think it's the county, pays for health club membership for people with medical conditions, and that they'll actually pay for a stationary bike not a Peloton, but a lower end, stationary bike for people to have at their home as well. And so it just made me wonder whether there's something like that we can do here also. And why care about anxiety and depression? This is another study from that was presented at CROI from the NA Accord. And as I always say, when it comes to NA Accord and some other studies, with great power comes great responsibility. So they had over 33,000 participants with over 160,000 person years of follow-up, and still only had 869 total MI events. So very relatively small number for this degree of follow-up. And luckily, Emily Heil, the first author, reminded me of the difference between a type 1 and type 2 myocardial infarction here. But what they found was that depression was associated with both types, and anxiety was associated with type 2. And these are the type 2 MIs down here. You can see anxiety, the adjusted hazard ratio was 1.42, and for depression, it was 1.20. So it seems to be a reason to care about anxiety and depression over and above that they are negative things for our patients. So Andrea did share this slide with me, but just transitioning to also co-infections. So several of these, I'm a little proud to say, are from our group: Christina Mara, Luminita Eddy, Ajay Bharti, myself, Dr. Janella, Mattia Trunfio, but showing that many co-infections are associated with worse neuropsychiatric outcomes in our patients. And some of these won't be surprising, but still the herpes viruses, I think, are very important because we can treat this. Well, that doesn't go away so much, but it's mostly latent. This usually goes away, but we can have long COVID. But we're working more on herpes viruses, and we have this publication from 5 years ago just showing that in people on suppressive antiretroviral therapy, the higher your CMV IgG, the worse your global performance. And we think that's because your people are having a greater immune response to CMV reactivation, CMV replication. And Dr. Janella has done a lot of nice work in this area and has a clinical trial in the ACTG for anti-CMV therapy, letermovir. So please refer people. This is not cognitive impairment only. It's just people with HIV and CMV on suppressive antiretroviral therapy. But at this year's CROI, Sarah and Katie Riggs, one of our infectious disease fellows, and myself and others reported on work that Sarah's lab did measuring CMV DNA and EBV DNA and EBV IgG and CMV IgG, but finding a sex-specific effect between CMV DNA and neurocognitive performance. And that's shown graphically here for global mean T-score. The women, the lower the CMV DNA, the worse the neurocognitive performance. And we were scratching our heads about that because we didn't expect it to be lower, but we think it makes sense because of the CMV IgG, that if you have more CMV replication, you have a greater CMV IgG immune response, which then reduces the DNA. So we thought that this might be reflective of CMV IgG. And in fact, in the CMV IgG that Sarah's lab measured, they did see, again, only in women, that women who had undetectable CMV DNA had higher CMV IgG. So there seems to be something here. Again, we're doing a clinical trial. Please refer people. But I'll just want to transition now to neurotoxicity of antiretroviral drugs. I'm not going to go through this in great detail. You guys know about efavirenz. You know about the vascular effects of protease inhibitors. You know about the data on dolutegravir that I previously mentioned. And so to get at this further, I wanted to just provide an update about our 19-fluorine MRS study. So this is a method that instead of doing magnetic resonance imaging or spectroscopy with a proton with 1H, as we usually do in the hospital and clinic, we use fluorine. We don't have to administer anything, but this detects fluorinated compounds in the body. And many of the antiretroviral drugs like FTC are fluorinated. And so this was used in the past to look at fluoxetine and here fluvoxamine, to measure their concentrations in brain tissue. So we got an R21 to try measuring dolutegravir, FTC. We looked at doravirine. This was very badly affected, this project, by the pandemic. We still got some people in, couldn't detect anything. We could find the drug on NMR spectrum in the lab. We couldn't find anything in the brain tissue. And luckily, instead of concluding that there was nothing there, which we didn't really believe, our imaging scientists said, you know, it's very possible that the drug is bound to something in the brain. And so let's dose some mice and collect their brains and put them in the NMR spectroscope in the lab and see if we can detect it. And in fact, they did, but you can see here it shifted. We would have expected doravirine to be here, which is where we were looking in the people, but it's actually here. And so, you know, we've learned something, and now we have to go back to NIH and say, you know, can we have some more funding to do this work in animals, and then figure out where this peak is in animals, and then go look for it in people. So we still hope that we can measure fluorinated antiretroviral drug concentrations in brain tissue, but you know, when you fail or make a mistake, you learn something, and we learned something from the R21, which is what R21s are in part meant for. So for neurotoxicity, we don't need to stick to antiretrovirals. So benzodiazepines, anticholinergic burden, and polypharmacy, oh my. So here's the cowardly lion on here for you. But Rowan Saloner, who's one of our really productive former postdocs, who's now up at UCSF, did some very nice work with Mariana Cherner and Aaron Sunderman, looking at the influence of benzodiazepines. And again, I won't take you through all this graph in the interest of time, but Rowan, not surprisingly, found that people who use benzodiazepines performed worse on neuropsychological testing. And Aaron went back, Dr. Sunderman, one of our faculty here, and looked at the relationship, the interaction with a medical burden index that they calculated, and showed that it was particularly in these people who were using benzodiazepines that the effect of the medical burden index was most prominent. So the greater the medical burden, even if you don't use benzodiazepines, the higher the probability of neurocognitive impairment. But if you use benzos, that's really amplified. And so we've been looking at this more recently with polypharmacy in general, so concomitant medication, number of concomitant medications here, as well as anticholinergic burden with Sante Kamkwalala in our STAR program and other STAR scholars. And we reported this at CROI, but we finally got this manuscript circulated to co-authors last week. But this is just, again, essentially correlation coefficients, standardized beta effect sizes and p-values, looking at not only global neuropsychological performance, but performance in all of these 7 cognitive domains that we assess, as well as the Beck Depression Inventory. And essentially what we found was that people who use more concomitant medications, who have polypharmacy, perform worse on neuropsychological testing, and people who have a greater anticholinergic burden have more depressive symptoms, even after accounting for the effects of the underlying diagnoses for which those medications are prescribed. Now, you know, I don't have that many slides, but I'm a talker, but I did want to talk about biotypes a little bit, and I'm just gonna, with apologies, I'm gonna have to breeze through these, but this has been a big movement in the field to try to bring together not just the one part of what the brain does, the cognition, but the depression, the anxiety, activities of daily living. And so I won't walk you through this slide. This is just to say that this has been being done in psychiatry. And so there's a lot of effort to do this. This is analysis by Raha Dasgeb at Hopkins, who's an amazing analyst and expert in machine learning. And what Raha was working with some of our investigators and investigators at Hopkins, like Leah Rubin, was some of the earliest analyses of these domains, these cognitive abilities that we measure, to let them tell us what the groupings are, rather than beforehand saying, this is how we put together these cognitive abilities and this is how we think they should be. Let's look at the data and let the data tell us. So what Raha found was that there was a group that had poor verbal fluency, another group that had poor executive functioning, poor learning and recall, and poor motor function. So it's a little bit different than how we usually look at this. And the intent is that, our clinical trials have not done well. Maybe we don't understand the biology as well as we'd like to think we do. Let's look for different ways of understanding these data that might be more strongly linked to biological mechanisms, so that we might be able to devise therapies that'll actually help people. We've been doing this in Charter. We reported this at CROI and have a manuscript submitted to neurology that's under review. But essentially, I won't walk you through these, but we took these cognitive abilities, added in the Beck Depression Inventory subscales, as well as the patient's assessment of own functioning. These are cognitive symptoms, as well as activities of daily living. So we expanded it to 17 different measures, working with Bin Tang, one of our wonderful analysts here. And again, we found that the data combined in a way that was different than our usual expectations. And I can walk you through these, but I won't in the interest of time. And that when we look over time, these cross-sectional phenotypes differ with how people do over time as well. So we're continuing to work in this area. We think it's going to be fruitful. Rob Paul from the University of Missouri at St. Louis has done similar work looking at longitudinal cognitive trajectories in his SABIS cohort in Peru. And the nice finding here is not only that the one finding is that Rob found 5 different groups of people depending on where they started out and how they did over time. The nice finding is that after people went on therapy, everyone improved into the normal range over 192 weeks of follow-up. So some encouraging news. And on that front, here's my EOR. They don't always have to be so negative. Rowan Saloner, again, has been really focusing on super aging. We tend to focus down here on the cognitively impaired CI people in the white, but Rowan's looking at that other end, perhaps the people with undetectable CSF viral loads, but there's a group of people who do well. And this is just showing that they have better cognitive reserve and better physiologic reserve. And, we can talk about how Rowan calculated that cross-sectionally. And then here in this more recent publication, looking longitudinally, these people seem to not only do well at the earliest time point, but over up to 5 years, seem to continue to do well. So there is a group of people with HIV who do well over time, and it seems to be linked with cognitive reserve, having better cognitive abilities before they get HIV, as well as better physiologic reserve. Again, I mentioned about hopefully these new phenotypes will be better linked to indicators of biology. We did do a genome-wide association study in Charter. We didn't find any statistically significant associations with our usual way of looking at cognition. We did with these new cognitive phenotypes, or the phenotypes that Ben has been looking at. So some early indication that they may be linked better to biology. This is some analyses I did yesterday using a new O-Link instrument that the San Diego CFAR purchased for us, in which we can measure group panels of 92 biomarkers. And in this heat map, I just won't go into great detail. I just want you to notice that using this cluster analysis, you can readily see there's this one group in blue where almost all the biomarkers are in the lower range, and this one group in red in which nearly all the biomarkers in the red range. And this is a way of just graphing that out. Here's the high biomarkers, here's the low biomarkers, and these differ in this old way of looking at it. I didn't have the clusters readily available to me when I was doing this yesterday, but if we just look at global impairment, these people with the higher levels of all these biomarkers, 63% increased relative risk of impairment. So more to come on that regard. We're out of time, so I'll skip this more cool findings on biomarkers from Charter. These are data from our colleagues in Gothenburg, Sweden. Magnus Gislin has published a lot in this area and basically was making the point that as people's CD4s decline, the pattern of biomarkers changes as well, which is just again another complexity of trying to figure out, what the combination of biomarkers are that indicate risk for having more neuropsychiatric complications. And then we're almost done here, but we're also doing machine learning of the prescribed drugs, the concomitant medications as well, with Raha at Hopkins. And Raha in this group, this is a different group of people. She found 6 different phenotypes, then we put a lot of work into categorizing the drugs, and she did principal components analyses. So these are rotated components, RC, of the different drugs. And you can see here in global impairment, rotated component 1 is important here, and this phenotype, and not surprisingly, psychotherapeutic drugs, CNS drugs, anxiolytics and sedatives, antipsychotics, benzodiazepines, antidepressants. So consistent findings, if not surprising findings, about what might be contributing to these problems. So interesting data on brain age gap. I thought I was not going to talk too long this time because I have like 37 slides, but some cool data on brain age gap. Won't get to talk about machine learning for imaging data, work we're doing in Charter. Please refer to our new Charter Plus cohort. We're enrolling people with HIV and without HIV now. So please refer people. And if you're interested in more about the biotypes, this is a big effort by NIMH to do this. So we published articles on this in JID. And so just refer you to these if you want to read more about that. And again, please refer, please refer, please refer. So sorry for going long. I thought, as I said, I thought I wouldn't, but oh well, lot to tell you.
Great. Thank you, Scott. We don't have too many questions, just a very few. I think Jeff asked, in Charter, did they measure past psychological trauma as well? Do you know?
We're doing that now, Jeff, because there's, as you know, there's so much data emerging on current stress, but also past life stress, particularly childhood trauma and sexual violence and so on. So we are collecting that now in Charter, but we didn't have the foresight, 20 plus years ago, unfortunately, to do that.
Okay. Robert Dice asked, this was regarding the methamphetamine and cannabis study. I'm not sure exactly. He said, is it because they use less methamphetamine if they were also cannabis users? I'm not sure.
We do collect those. Yeah. So I mean, people differ in the amount of methamphetamine they use. I should say these were all former methamphetamine users, so they met criteria within the last year for methamphetamine dependence or abuse, and they had been abstinent for at least I think 4 weeks prior to the assessment, and so they were not actively using, but so it speaks more to the chronic effects of meth. But we do have data on the extent of meth use, and I just don't recall them, Robert. I'm sorry about whether those two groups differed in terms of the extent of their prior use. I think maybe the non-cannabis users maybe started using cannabis at a younger age, but that there weren't many other differences between them.
Okay. And then last, I think it's a comment or a question from Gigi. Did Denver pay for it from tax money generated from cannabis sales? This is your exercise study.
Yeah. No, we'd have to ask Christine. I'm not sure it's known, but it's a nice idea. We ought to make the state pay for it from tobacco taxes, but But yeah, I mean, it's one way of thinking about how we might convince the state or the county to fund something like that, right?
Yeah. Jeff Taylor says Christine got grants, not cannabis tax money. So,
Right.
all right. Well, thank you, Scott, for an excellent presentation. And thanks everyone for being here. And Gigi, yeah, I figured it was a joke. And I think we are off for the month of July. We will be back in August. So have a good break, and we will see you back in August. Thanks, everyone.